Hi everyone. This is the last respiratory physiology lecture, and then there will be one blood lecture after this. I'll try to get them all up. They usually would go up on Monday. That's when I usually do the last lecture, but I'm going to try to get them up before the weekend so that you guys can have some time to work on it over the weekend. Your test will be next week. In the announcements, I posted the dates and times. So make sure to go back and look at that. Oh, no, that's good. Okay, so where we were last time was we watched this video at the very end, and this video is kind of where we're going to start today. So I'm just going to play it one more time. I won't interrupt it. And then the objective today is to figure out what happens with specific gas exchange, not air, that's ventilation, but gas exchange, which is respiration, at the alveoli versus to the pulmonary capillaries, and then between the systemic capillaries and the tissue cells. After we figure out what's going to happen, I want to talk about some things that make it more likely whether you will pick up oxygen or drop off oxygen, and the same with carbon dioxide. And then the very last thing in this set of notes is just to investigate the things that change your ventilation rate or your breathing rate. Okay, so let's just watch this one more time.
Fresh air entering the lung carries oxygen with a PO2 of 160. The presence of moisture in the lung results in reduction of the PO2 to 104. Fresh air entering the lung carries carbon dioxide with a pCO2 of 0.3. The carbon dioxide delivered to the lung from the blood raises the pCO2 to 40. At the arterial ends of the pulmonary capillaries, oxygen diffuses from the air in the alveoli into the blood, and carbon dioxide diffuses from the blood into the alveoli because of differences in partial pressures. As a result of diffusion, at the venous ends of the pulmonary capillaries, the PO2 in the blood is equal to the PO2 in the alveoli, and the pCO2 in the blood is equal to the pCO2 in the alveoli. With no differences in partial pressure, there is no more net movement of oxygen or carbon dioxide. Oxygen diffuses out of the arterial ends of tissue capillaries into the tissue fluid, then into the cells, and carbon dioxide diffuses out of the cells into the tissue fluid, then into the blood because of differences in partial pressures. At the venous ends of tissue capillaries, the PO2 in the blood is equal to the PO2 in the tissue fluid, and the pCO2 in the blood is equal to the pCO2 in the tissue fluid, resulting in no more net movement of oxygen or carbon dioxide. The blood now carries the oxygen and carbon dioxide to the lungs. In the body, all of these exchanges occur simultaneously.
Okay, so pull up one more thing. All right, so I'm just going to go through this in the notes. It's the same concept that we've looked at before. So when we look at external respiration, please remember that when we're talking about external respiration, we're talking about all of the gas exchange until you get to cellular respiration. So really this whole picture is external respiration. Cellular respiration is considered internal respiration. So external respiration is gas exchange two different times. It's gas exchange right here between the alveoli and the pulmonary capillaries, and then between the systemic capillaries and the cells. Those both are steps of external respiration. So the general information about how gas exchange goes is that gases are going to move first here across what we call the respiratory membrane. So let's look back at that for just a second. The respiratory membrane is this very thin, generally just 2 layers of simple squamous epithelium with a basement membrane in between. This very thin layer that you're looking at right there, and it's about 0.2 to 0.4 micrometers, 1/6000th of an inch thick, so really, really thin. In addition, there's tons of it, because if you spread out the surface area of both of your lungs, you have about 300 million alveoli in healthy lungs with a combined surface area of about 750 square feet of respiratory membrane. I've lived in apartments smaller than that, 25 feet by 30 feet. The thinness and the large surface area allow for really, really efficient diffusion of gases. In addition, your lungs have a disproportionate amount of blood in them at any given time. You've got about 5 to 6 liters of blood, and at any given time coursing through the vessels and capillaries of your lungs, you have about 1 liter of blood at any given time. Okay, so the respiratory membrane is this that we're looking at right here, but let's look at it in chapter 17. So basically what we are going to see that occurs at the respiratory membrane is gases are going to move down their partial pressure gradients, which is kind of the same, again, as saying that they will move down their concentration gradients. So oxygen is going to move from high oxygen in the alveoli to low oxygen in the blood. Okay, so from high to low. And then carbon dioxide is going to move from relatively high oxygen in the blood that was delivered to the lungs to the relatively lower oxygen in the alveolar airspace. And how does that occur? Generally speaking, this is going to occur just by simple diffusion because the solubility of oxygen and carbon dioxide. So the respiration process, and remember respiration means gas exchange. The respiration process of in the lungs is that the blood that's coming into the lungs via the pulmonary arteries was low PO2, deoxygenated. It's never 0 oxygen, by the way. It's just low oxygen versus high oxygen, and high PCO2. And so the circulation is going to move the blood from the pulmonary trunk to the pulmonary arteries to arterioles, capillaries, all the way down to those capillaries that are covering the surface of each of the alveoli. And what is going to happen is as the blood moves by the alveoli, things are going to move down their partial pressure gradients. And then I'll talk about this in more detail later. But what happens, of course, is the oxygen not just moves into the blood plasma, it actually moves into the red blood cell and takes a ride on hemoglobin, which, of course, when oxygen binds to hemoglobin, it makes binds to the iron, the heme portion of hemoglobin. It makes the blood bright red. So typically speaking, if you are looking in a healthy person and you're looking at pulmonary venous blood that's on its way back from the lungs to the heart, it's going to be bright red. And then systemic arterial blood is also going to be bright red. Blood is not blue, despite the fact that my son doesn't believe me that blood is not blue. It's red, bright red, and dull red. Okay, so that's what I was meaning about the loading. So it has to load onto hemoglobin. And then carbon dioxide is going to do the opposite of what we just said. Carbon dioxide is going to diffuse from the blood as it came back from the pulmonary arteries, ultimately to the lungs. It was relatively high carbon dioxide. It is going to diffuse into the alveoli, and then you can breathe it out. Of course, circulation is going to follow. Now you've got the blood that has just gone by the alveoli and its way back into the pulmonary veins, and now that blood is going to be nice and rich in oxygen and bright red and low in carbon dioxide. We're going to send it back to the heart, back to the left atrium actually of the heart, into the left ventricle, and then we're going to send it out into systemic circulation. So all of this probably makes perfect sense to you guys. So what's going to happen when you send it back into systemic circulation is kind of exactly the opposite. We don't have any alveoli to deal with it, of course, but the blood that's coming from the lungs goes into the left side of the heart and then gets pumped into systemic circulation. All of the blood here is going to be relatively high PO2 and relatively low PCO2. And of course the tissue cells, because they're constantly working, are going to have low PO2 because they're using it up, and high PCO2 because they're generating it, both from cellular respiration. And what is going to happen is, of course, the oxygen that you bring down here is going to have to jump off of the hemoglobin bus before it can actually diffuse into the cells. So it's going to get off of hemoglobin from the red blood cells, and it's going to diffuse from the blood to the interstitial fluid into the tissue cells. And then the carbon dioxide that was generated by the tissue cells is going to diffuse the opposite direction, right? And all of these have all of the amounts on there. So I am going to diffuse from the cells to the interstitial fluid to the blood plasma with carbon dioxide. And then, of course, circulation is going to follow again. So we are going to take this relatively low oxygen blood that will now be dull red because there's not as much bound to hemoglobin. And then I am going to deliver it back to the right side of the heart, and then I'm going to start this process all over again. Okay, so all of this to say that what we just talked about was basically the process of external respiration. And external respiration happens first at the pulmonary capillaries and then at the systemic capillaries. But again, like we talked about last time, it is all really serving cellular respiration, which is the thing that is constantly using up oxygen and generating carbon dioxide. So cellular respiration is really responsible for these gradients that the rest of it is diffusing down. Okay, and these processes all happen by simple diffusion, so you don't have to worry about transport mechanisms or anything when you get to them. Okay, so now what we get to is the part that gets really confusing at first, so just give me a minute, and by the time we're finished, I will put it all back into a recap that will make sense. This is easier to teach in person. We can do this, though. So I want to talk about how gases are transported in the blood. Now that we know, we pick them up, we take them from point A to point B, but how do they ride? So when I'm talking about oxygen transport, a little bit of the oxygen is always still in the blood plasma, kind of on its way to and from the red blood cells. But that's only about 1.5%, and it adds directly to the PO2. But then the rest of it, is this the right figure? Oh yeah, the rest of the oxygen, generally speaking, around 98.5%, combines with the hemoglobin. Okay, when it combines with hemoglobin, and we'll talk about hemoglobin in a little bit more detail in the blood notes, which are right after these. But hemoglobin is basically 4 amino acid chains, 2 alpha, 2 beta, each of which has a heme group in the middle, and the heme group has iron. And so the globin portion is the protein portion. It's a glob-shaped protein, hence globin. And then the heme portion is the iron-containing portion. And in each, this is just like a diagram to kind of show you what it means. Okay, so this is a diagram of like a physiological diagram of hemoglobin. So there are 4 subunits, each of which has a heme group. Each heme group can carry an O2 molecule. Okay, so iron sits on the heme seat. Okay, keep that in mind. So oxygen rides on the heme seat. So if hemoglobin is a bus that carries oxygen, then it carries gases actually, because it can carry carbon dioxide, then it's got 4 seats that are reserved for oxygen. And we'll talk about how it carries carbon dioxide in just a second. So iron combines with oxygen to form hemoglobin. And so this is a loose reversible interaction. A bus doesn't do you any good if you can't get on it and can't get off of it. So this combination is loose and reversible. Oxyhemoglobin tends to be bright red in color, and deoxyhemoglobin is not bright red in color, it's duller red in color. Okay, so now what we need to do, and this is the part where it gets kind of confusing for a minute, and then I'll be able to bring it back together. So what I want to do is I want to talk about what determines how much oxygen the hemoglobin bus is carrying at any given time. Okay, so I want to talk about the factors that determine how much oxygen combines with hemoglobin. Okay, the other way to say that is what is hemoglobin's affinity for oxygen, or how saturated is hemoglobin with oxygen. Okay, so this hemoglobin is 98.5% saturated. There's only one seat that's open. But this hemoglobin is 75% saturated. Cool? Okay, so the first thing I want to talk about with that is, so what determines how much oxygen is bound to hemoglobin? There's several factors. So the way that I think it's really useful to think about this is to think about what should be true based on your experience about pulmonary capillary beds versus systemic capillary beds. Okay, so if we imagine that with pulmonary capillary beds, what should be happening, so stick with me for a second, what should be happening is that pulmonary capillary beds, you should be loading oxygen onto the hemoglobin bus. You should be loading, right? So everything that happens at pulmonary capillary beds should favor loading, right? Okay, so you need to carry it around. And then what should happen at systemic capillary beds is that everything that's true at systemic capillary beds should favor unloading of oxygen, because oxygen can't get into, for instance, the tissue cells unless it gets off the bus, right? So it can diffuse out. So one more time, everything that is true at pulmonary capillary beds should favor loading oxygen onto hemoglobin. And everything that is true at systemic capillary beds should favor unloading oxygen off of hemoglobin. So you load, right, to carry yourself to work, and then you unload because you've got work to do. Cool? Okay, so let's talk about the factors that determine how much oxygen combines with hemoglobin. So the first factor is kind of simple, and it should be relatively logical to us. And that is, one of the things that determines how much oxygen loads onto hemoglobin is how much oxygen there is. Okay, so the PO2. So what is true is if PO2 is higher, it is more likely to load onto oxygen. If PO2 is lower, it's less likely to load onto oxygen. So if you look right here, and these are systemic, by the way, higher PO2, higher saturation, lower PO2, lower saturation. That's kind of just the law of statistics and averages. So the higher the PO2, the more oxyhemoglobin is formed, and vice versa. This is called saturation. So higher PO2 causes loading of oxygen onto hemoglobin for transport, and that is true at the lungs. So let's compare. If you had to compare pulmonary capillary beds to systemic capillary beds, which one would have a higher PO2, generally speaking? Pulmonary or systemic capillary beds? Pulmonary capillary beds, right? They would have a higher PO2 because they're bringing in a lot of oxygen, whereas systemic capillary beds would be, they would have used a lot of the oxygen. So higher PO2 favors loading of oxygen onto hemoglobin for transport. So why is it so good at loading? Why do we generally get about 90% saturation almost all the time? It's because there's such a good gradient between how much oxygen is in our atmosphere and how much oxygen is in the blood going through the lungs. So in lungs, hemoglobin is about 90% saturated, even when our PO2 is only 60 millimeters of mercury, because the air is generally 104 millimeters of mercury of the PO2. So the saturation is relatively easy. I don't know who's calling me. I'm going to ignore that. Okay, so the opposite is also true though. Wherever there's a low PO2, you are relatively likely to get off the bus. Okay, so high PO2 favors loading, and higher saturation. Low PO2 favors unloading and lower saturation. So that's a good place to start, right? Hopefully that one really makes sense to you. Okay, because now what we're going to have to do is we're going to have to get into the ones that might make a little less sense, but stick with me, we'll be able to do it together. All right, so the next one is pH. So before we start to predict, I want to try to remind you guys of this damn equation that you've seen 3,000 times. Okay, so this is the important equation. So as carbon dioxide increases, H plus tends to increase and pH tends to go down. So with that in mind, which capillary bed, pulmonary versus systemic, do you imagine will tend to have a lower pH? Okay, again, as carbon dioxide goes up, H plus goes up because of this damn equation, the bicarbonate equation. And as H plus goes up, pH tends to go down. So the pulmonary capillary bed, I'm going to make this one up here, closer to the lungs, and then the systemic capillary bed. Which of these two capillary beds do you predict will have a lower pH? I predict that the one with more carbon dioxide, meaning the systemic capillary bed, will have a lower pH, and I'm right. So in low pH situations, basically, are you more likely to get on the bus or off the bus in low pH situations? Well, what do you want to do in the systemic capillary beds? Low pH favors unloading. A higher pH favors loading. Okay, just comparing the two capillary beds to one another. So basically it says as your pH drops here, the affinity of oxygen for hemoglobin also drops, and you're more likely to get off the bus. You decrease the affinity. As you increase pH, your affinity of oxygen for hemoglobin increases, and you're more likely to get on the bus. So in low pH or acid situations, hemoglobin more easily gives up oxygen, so getting off the bus at low pH situations. The acidity occurs in the tissues for a bunch of different reasons, but this is certainly one of the big ones. So here's the prediction. If it's low pH, as is true in systemic capillary beds, it favors unloading of oxygen from hemoglobin because you're going to use it in cellular respiration. So get off the bus to go to work. And if it's higher pH, as is true in pulmonary capillary beds, it favors you getting onto the bus, the hemoglobin bus, to go to work. Okay, so we've got these two now. We have a prediction about PO2 and we have a prediction about pH. Okay, and then PCO2 and PCO2. So with PCO2, really just figure out which capillary bed has a higher PCO2, and the capillary bed with a higher PCO2 is going to give you the prediction of the next part. So which capillary bed, pulmonary versus systemic, is going to have a higher PCO2? Okay, systemic capillary beds, right? So an increase in PCO2 should do what you need to do at systemic capillary beds. And what do you need to do? Do you need to get on the bus to go to work, or get on the bus to ride to work, or do you need to get off the bus to go to work? So you need to get off the bus, right? You need to get oxygen off of the hemoglobin bus because you need to use it for cellular respiration. So not surprisingly, an increase in PCO2 causes hemoglobin to let go of oxygen, like in systemic capillaries. So does that cause what goes here? An increase in PCO2 causes loading, getting on the bus, or unloading, getting off the bus? Unloading, right? So you're going to get off the bus, let hemoglobin off, like here, let hemoglobin, let oxygen off of the hemoglobin bus so that you can actually use it for cellular respiration. And the opposite is also true. A decreased PCO2, as is true in pulmonary capillary beds, is more likely to make you load. Okay, and then only two more things, and then we will be ready to do a summary. Okay, so temperature is the next one. Which capillary bed do you imagine is generally warmer? The pulmonary capillary beds or the systemic capillary beds? What do you think? And also, is a capillary bed warmer when it has higher metabolism or lower metabolism? That's also true. Okay, so if you compare pulmonary capillary beds up here to systemic capillary beds down here, generally speaking, the pulmonary capillary beds are going to be cooler and the systemic capillary beds are going to be warmer, and they will get even warmer as they do higher rates of cellular respiration. So with that in mind, predict what temperature is going to do. Oh, I lost my two. No, I can't make that subscript. Okay, so as you increase your temperature, like comparing pulmonary to systemic capillary beds, when your temperature goes up, as could be an indication of higher metabolism, do you want for oxygen to get on the hemoglobin bus to ride around, or get off the hemoglobin bus to be used? Okay, so your prediction, hopefully, is that an increase in temperature causes oxygen to get off the hemoglobin bus. Oh, that is in the wrong place. To fix this, I must have just accidentally cut and pasted that on accident. I'll fix it. Okay. All right. So here's the prediction: an increase in temperature. Oh my goodness. An increase in temperature. Sorry, that's distracting as hell. An increase in temperature does what? Loading or unloading. Increase in temperature favors loading or unloading of oxygen from hemoglobin. So again, just compare the pulmonary capillary beds to systemic capillary beds. The systemic capillary beds are hotter, so an increase in temperature is going to favor unloading, because that's what you need to happen at the systemic capillary beds for use in cellular respiration. And of course, this occurs in tissues when they're more active. It also occurs with exercise. So basically, if a tissue becomes more active, you're more likely to release the oxygen into the tissue for use. As you're exercising, you will release more oxygen. As your body heats up, you will release more oxygen. All of those things are true. Okay, and then the last one with oxygen is the one that's just a little weird to describe, but I can do it with you. And that is this one. Okay, you haven't heard of this before. This is BPG or DPG. It's 2,3-diphosphoglycerate or 2,3-bisphosphoglycerate. Unfortunately, they haven't gotten their crap together about which one. Okay, so here's the deal. Just listen for a second. Red blood cells don't need oxygen for their cellular respiration because they don't have any mitochondria. Okay, so what happens in red blood cells is their only source of ATP, and they don't need very much of it because they're not very active except as like little buses. So in red blood cells, during glycolysis, they produce this byproduct called BPG or DPG. And as the rate of BPG or DPG increases, the red blood cells kind of use it as an indicator of what is going on with other cells. They say, hey, if I'm doing a lot of ATP production, then maybe other cells are also doing a lot of ATP production. And if that is true, maybe I should let go of this oxygen that I am carrying around to serve other cells, because they don't use the oxygen for their own cellular respiration. So here's the deal. With red blood cells, as they increase their DPG production, it will favor unloading of oxygen, because think of it as red blood cells using their own rate of metabolism as an indication of what is needed in other cells, and then taking one for the team and saying, okay, I realize you guys might actually need more oxygen, so an increase in DPG favors unloading of oxygen from hemoglobin for use in cellular respiration. And so what it does is it actually pushes oxygen out of the red blood cell. So an increase in DPG occurs when metabolic, I don't know how the hell I did all these, I accidentally did all this, metabolic O2 an increase in DPG when metabolic O2 increases. And it moves it off of hemoglobin. It decreases the affinity for hemoglobin. All right, so here's the summary. And there's also a good summary right here for those of you who are flowchart people, 1713 right here. This kind of shows you all, no, that's carbon dioxide, 17, I don't know where it is. Oh yeah, that is it. Okay, so here's the thing. So one more time. So what we really need to do, the best way to remember this that I've ever found, is to figure out what you think is true at systemic versus pulmonary capillaries. Okay, and then make everything that happens at a systemic capillary favor what you want to occur at a systemic capillary, which is pulmonary capillary, which is everything that occurs at a pulmonary capillary favors loading, and everything that occurs at a systemic capillary favors unloading. Again, the metaphor is this. Loading means you're getting onto the bus, right? Oxygen is moving on to hemoglobin so that you can take it to work. Unloading, as is true at systemic capillaries, means you are getting off of the bus, oxygen is, to go to work during cellular respiration. So let's see what is true comparatively in systemic versus pulmonary capillaries. Everything that happens in a systemic capillary should favor unloading, yes? So what happens in systemic capillaries is comparatively there is a lower PO2 and a lower pH. The lower pH is usually due to carbon dioxide. There is a higher PCO2, comparatively higher temperature, and as the tissue gets more active, a higher BPG. All of those things are true at systemic capillaries, and so all of those things should favor unloading, right? Letting oxygen off of systemic capillaries. In pulmonary capillaries, comparatively, just compared to systemic capillaries, higher PO2, relatively higher pH, a decreased PCO2 because you're breathing out carbon dioxide, lower temperature, and assuming the metabolic rate is a little lower, lower BPG. And all of those things are true at pulmonary capillaries, and they should favor oxygen loading onto hemoglobin. Cool? Okay, one last little tidbit about that before we go into carbon dioxide, and that is, what about carbon monoxide? Carbon monoxide is really interesting because carbon monoxide is rude in that carbon monoxide sits in oxygen's seats on the hemoglobin bus. Carbon dioxide does not do that. Carbon monoxide has a greater affinity for the heme group in hemoglobin than does oxygen. It sits in the bus and it doesn't get out of the seat. Every single time you go around, if you are, for instance, breathing in carbon monoxide, it will take up a seat, and then the next time it will take up more seats and more seats and more seats, and it doesn't really let go of the seats. So what's going to happen is as the blood goes around through the lungs and you're breathing in carbon monoxide, it takes a disproportionate amount of seats on the hemoglobin bus, and eventually you're carrying more carbon monoxide than you are oxygen, and your blood will be bright red because the oxygen in carbon monoxide also makes blood red, but you will be carrying almost no oxygen. So it's called hypoxemic hypoxia. It's not easily reversible. It displaces oxygen. It will definitely kill you if you don't get out of it, or if there's no intervention before you're almost all saturated with carbon monoxide. It's really, really dangerous. And of course, carbon monoxide, a lot of people misunderstand this, and I know some of you guys know it, but I'll just go ahead and tell you. Almost everybody in Utah has homes that are heated by some kind of carbon-based, like natural gas, for instance. And carbon monoxide is colorless and odorless, and it's a byproduct of burning carbon-based fuels. So car exhaust, tobacco smoke, charcoal smoke, natural gas, all of those kinds of things. Now, the natural gas that heats my home, before it's burned, of course, it has a smell, right? And the smell was put in there so that you could tell when it was leaking. That's not what carbon monoxide smells like. It's colorless and odorless after it's burned. So if, for instance, the exhaust system in my heating unit is leaking, then it will leak the byproducts of burning that gas into my home. And it's colorless and odorless, and you just kind of get exhausted because you're not getting enough oxygen around, and then you fall asleep. And if there isn't fresh air coming in and or intervention, you just don't wake up, especially with little ones. So the reason this is worth mentioning is because we're getting closer to the time in which, for instance, people are going to be using barbecue grills. Don't use them in your garage. Don't turn on your car exhaust in your garage. And of course, you should have carbon monoxide detector in your house if it's heated by any kind of carbon-based burning product. Okay, so now I want to talk about how you carry carbon dioxide around in your body. And carbon dioxide is, of course, a byproduct of cellular respiration. So how do you carry this around in your body? And this looks really hard at first, too, but it gets better, I promise. Okay, so basically, with oxygen, you either carried around 98.5% of it in systemic arterial blood on the heme portion of hemoglobin, and then 1.5% of it was at any given time dissolved in the blood plasma. But carbon dioxide is really different than that. So I want to talk about proportions of carbon dioxide. So first off, carbon dioxide travels in the blood in 3 different forms, and we're going to go through this a couple of times, so don't worry about it the first time through. At any given time, about 7% of the carbon dioxide is just in the blood plasma as carbon dioxide, like in the blood plasma. And that adds directly to your PCO2. But 23% of your carbon dioxide actually diffuses into the red blood cell and sits on hemoglobin. But importantly, it does not sit on oxygen's seats. It combines not with the heme portion of hemoglobin, but with the globin portion of hemoglobin, okay? And so it doesn't change the color of the blood or anything, and you can carry it simultaneously when you're carrying oxygen because it's not sitting on the same seat. But this is it right here, combines with the heme portion of hemoglobin. But 70% of carbon dioxide diffuses into the red blood cell, and then the red blood cell forces a chemical reaction that you've learned before so that you can carry boatloads of carbon dioxide with really, really minor changes in pH. So here is what is going to occur. So 70% of the carbon dioxide will diffuse into the red blood cell and then gets converted into the bicarbonate ion. And this is the equation that you are doing. So what's going to happen in systemic capillaries is you are going to force this equation this way, but why would you want to do that? You're going to do that because then you can pick what you want to do with the bicarbonate ion and pick what you want to do with the H+ ion. And if you get to pick where those go, you can have a tremendous amount of control over pH changes. So this carbon dioxide is, of course, produced in tissues as a byproduct of cellular respiration, right? And it diffuses out of the cells and it moves into the bloodstream, okay? And as it moves into the bloodstream, again, just a little bit of review, 7% of it is just at any given time in the blood plasma. 23% of it is going to be loosely held onto by the globin portion of hemoglobin. But then 70% of it, you are going to force this chemical reaction. And so when I force this chemical reaction, it's a big deal if I can figure out what to do with the H+. Then I can carry boatloads of carbon dioxide with minor changes in H+. So in the red blood cells of systemic capillaries, you're going to force this equation this way. So you know it written like this, CO2 plus H2O via carbonic anhydrase, Ca, carbonic anhydrase, this one right here, goes to H2CO3, which dissociates to H+ and HCO3-. But then let's figure out what you do with those. What you do with those is the H+ also combines with hemoglobin, but a different portion of hemoglobin. It's got a positive charge. You can stick it onto a negative amino acid. And So basically now if H plus doesn't go into solution, it doesn't impact pH. So what I'm doing is I'm forcing this equation, I'm holding on to the H plus. And then what can I do with this bicarbonate ion? The bicarbonate ion, I can actually push out into the blood plasma so that it can have some buffering capacity in the blood plasma. And when I do that, in order to maintain the electrical gradient in the cell, I push negative ions out and I bring a negative chloride ion in. Okay, this is called the chloride shift. So I will go through this one more time with you in just a second. Okay, so let me show you that in the same figure. So if we are looking at the same figure, I'm going to go through all of this with you. Okay, so this is the equation. This is just a different way of looking at the same equation. So in systemic capillaries where I'm generating a whole bunch of carbon dioxide, I want to be able to carry as much of it as possible with as little impact on blood pH as I can. So I am going to carry 7% dissolved, okay? And then here I am going to carry 23% on the heme portion of hemoglobin. Oops, escape. On the heme portion of hemoglobin. And then 70% I'm going to do this with. Assume it had a percentage on it. Oh, let's see. Okay, so but of course you want to go through that thing again, and that thing is, okay, so the carbon dioxide, 7% of it stays here, 23% of it rides on the globin portion of hemoglobin, but then 70% of it does this. So same equation that we're looking at, CO2 plus H2O via carbonic anhydrase makes carbonic acid, right, H2CO3. And carbonic acid dissociates to the bicarbonate ion, HCO3 minus, and the hydrogen ion, which is H plus. So let's follow both of them. If I let H plus go into the blood plasma, I would really drop the pH dramatically, but I don't want to drop it dramatically because your blood gets to be about 7.34 to 7. I forgot. Your blood has a very narrow pH range before you start changing the structure of proteins. And then, so what am I going to do with this H plus? Well, I am going to hold on to it. I'm going to hold on to it with hemoglobin, okay? And it's not going to go into solution. What am I going to do with this bicarbonate ion that could be really helpful? What I'm going to do with the bicarbonate ion is I am going to push it out into the blood plasma to buffer whatever is changing the pH out there. But in order to maintain the electrical gradient of the cell, I'm going to do the chloride shift, which is pull that chloride ion into the red blood cell, okay? So this is all what happens at systemic capillaries. Now, to give you that as a start, now I want to figure out what's going to happen at pulmonary capillaries. So at pulmonary capillaries, we are going to kind of do the opposite, okay? So at pulmonary capillaries, what I am going to do is I am going to get rid of everything. We're trying to get rid of carbon dioxide, remember? And how am I going to get rid of it? Well, it's going to be relatively simple compared to what we just did. Okay, so the 7% that I had before that was dissolved in the blood plasma, that can just diffuse straight out because it's carbon dioxide, it's CO2, and that will just diffuse straight out. The 23%, the 23% right here where that was loosely bound with the globin portion of hemoglobin right here, that's going to be the next one that you get rid of. So you got rid of the 7%, the 23%. And then what we're going to have to do with the 70% is we were carrying it as the bicarbonate ion in the blood plasma, but you can't breathe out bicarbonate. So what you're going to have to do is you're going to have to take that bicarbonate and you're going to have to reverse everything that you just did to make it back into CO2, right? So what we did in the systemic capillary beds was move everything that way. In the pulmonary capillary beds, we're going to move everything this way. And we're going to do it by the same mechanism. So we're going to reverse the chloride shift. We're going to push chloride out and bring bicarbonate in, okay? That H+ that I was holding on to right here on my hemoglobin, I'm going to let go of that, right? And then the H+ and the bicarbonate ion, I'm going to push this direction and I'm going to make carbonic acid, and then carbonic anhydrase, that enzyme can actually dissociate. It can do both directions of that chemical equation. So carbonic anhydrase is going to dissociate carbonic acid to CO2 plus H2O. And now I have that 70% of the CO2 that I can breathe out. Okay, so I'm going to go through that one more time. Okay, so let's go back here. Ready? Probably don't want to do it one more time, but I'm going to do it anyway because it'll seem a little easier the next time that you watch it. Okay, so I'm at systemic capillaries. I'm generating carbon dioxide. I want to carry a lot of carbon dioxide without causing a major change in the pH. How am I going to accomplish that? I'm going to carry carbon dioxide 3 different ways. Some carbon dioxide I'm just going to allow to diffuse straight into the blood plasma. Right here in this carbon dioxide, 7% of it is just going to ride around as carbon dioxide in the blood plasma. 23% of it is going to hitch a ride onto hemoglobin. Okay, and then 70% of it is going to go through this equation. Can you see why I made you learn that equation at the beginning of the semester? Because it comes all damn semester. So then I'm going to do CO2 plus H2O via carbonic anhydrase goes to carbonic acid. Carbonic acid then dissociates to the bicarbonate ion and H+. I hold on to the H+ on hemoglobin so that it doesn't impact my pH much. I push the bicarbonate ion out into the blood plasma so that it can buffer whatever's there, and then I pull chloride back in to maintain the electrical gradient. So that's all happening at my systemic capillary beds, and now I'm going to take all of that carbon dioxide and I'm going to carry it back to my pulmonary capillary beds so that I can get rid of it. And I am going to do this process at the pulmonary capillary beds. So now I'm at pulmonary capillary beds. What's the easiest one to get rid of? The 7% that I already had right here. 7% of the carbon dioxide is breathed out. That was dissolved in the blood plasma. 23% comes next. That was bound to hemoglobin. You breathe that out. And then, of course, the 70% that you guys are tired of dealing with right now. The 70%, you reverse everything that you just did. You take the H+ off of hemoglobin. You reverse the chloride shift, push chloride out, bring bicarbonate back in. You combine those two into carbonic acid, and then carbonic anhydrase will dissociate them into H2O and CO2. And now you get to breathe out that last 70%. Cool? Not do you love it, because generally speaking, people don't. But do you get it? Okay, so now the only thing that we need to talk about is what determines, if this happens at systemic capillary beds and this happens at pulmonary capillary beds, what is it that determines which direction this equation goes, if it can go either direction? And really what it is just the concentration of carbon dioxide. If carbon dioxide is really high, which is called hypercapnia, it shifts the reaction to the right, which would be true at systemic capillary beds. If carbon dioxide is relatively low, which would be true at pulmonary capillary beds, then it's shifted this direction, okay? So now we come to breathing. So what is the primary way that you can alter PCO2 in the body? The primary way that you can alter PCO2 in the body is by determining your rate of ventilation. So if you increase ventilation, increase breathing, you are going to push off a whole bunch of carbon dioxide, hyperventilation. You blow off too much carbon dioxide. If, for instance, you get super stressed out and you hyperventilate for whatever reason, you blow off carbon dioxide. What that will do is if you blow off a whole bunch of carbon dioxide, right, you are going to decrease the amount of H plus in the body. So you got rid of too much carbon dioxide, and this can cause respiratory alkalosis because you're decreasing carbon dioxide, decreasing H plus, and getting more alkaline. But if you decrease ventilation, like with COPD, when you can't breathe well for whatever reason, you'll retain too much carbon dioxide, hypoventilation, right? And you'll increase your H plus. And this can cause respiratory acidosis, okay? So it just depends on how much carbon dioxide you're holding on to versus breathing out. This is why, by the way, your mom told you when you were hyperventilating to breathe into a paper bag, because, of course, when you are breathing off too much carbon dioxide, but then you breathe into a paper bag, you're breathing some of the carbon dioxide back in, right? Which helps to reset your pH. Okay, so last topic here, we did all the hard stuff, by the way, is control of ventilation. So back to the concept of breathing instead of just gas exchange and carrying gases around. So I want to talk about the concept of controlling ventilation. So here's the deal with how ventilation is controlled. It's primarily controlled by the nervous system, of course, neural control, and primarily by the somatic nervous system, because the somatic nervous system controls the diaphragm and the external intercostal muscles. So here's what happens with nervous control of inspiration. With nervous control of inspiration, what you are going to do is your inspiratory neurons are inherently excitable and rhythmic. The action potentials that will cause ventilation, inspiration, originate in the medulla oblongata and the pons. I don't know if you've ever looked at this before. I kind of talk about it in anatomy, but there are two respiratory centers, and I won't hold you responsible for the individual names of them, but the two respiratory centers, okay, are a respiratory group that's in the pons, and then some respiratory groups that are in the medulla oblongata. So those two work together to control ventilation. Okay, so these are inherently excitable and rhythmic, and what's going to happen is the medulla oblongata and the pons get to decide, I'll tell you how in just a second, when to send out a stimulus, and they will send it out via the phrenic nerves, right, to the diaphragm, and the intercostal nerves to the external intercostal muscles. And then what they are going to do is they are going to send stimuli for about 2 seconds, causing contraction of the diaphragm and the external intercostal. The size of the thorax expands, the pressure drops, and then air moves in because flow equals the delta P, the difference between alveolar pressure and atmospheric pressure over R. So air is going to move in because I dropped the pressure here compared to here. Okay, so that stimulus causing contraction of the diaphragm and the external intercostals lasts for about 2 seconds. And then, of course, the muscles involved in inspiration are going to relax, and your diaphragm relaxes and your external intercostals relax. And what happens, of course, is that decreases the size of the thoracic cavity, and since the lungs have elasticity, they will recoil. You decrease the size, you increase the pressure, and expiration occurs. That's passive expiration or exhalation. Okay, so what about your expiratory neurons? What do those do? So the expiratory neurons, they don't fire very often. Like normal tidal breathing, it's just inspiration caused by contraction of the diaphragm and the external intercostals, and then relaxation, which will cause expiration. So the expiratory neurons are going to not fire unless, for instance, you're talking or you're exercising. So they only send action potentials during high levels of ventilation, like I'm doing right now. And what they do is during high levels of ventilation, you will send an impulse to the internal intercostal via the internal intercostal nerves and to the internal intercostal muscles to force expiration by squishing your thoracic cavity, increasing the pressure a little further and causing expiration or forced expiration. So this is happening all the time, the inspiration, and it's muscular contraction. Normal tidal expiration is just muscular relaxation, but then when you're exercising or talking, you will use the expiratory neurons. Okay, so now let's talk about what can change your rate of ventilation. So what changes how quickly you breathe? Well, a couple things, and some of them will make sense to you immediately. One of them is like, since my cerebral cortex has the capacity kind of override my pons and my medulla oblongata up to a point, I can hold my breath. I can speed up my breathing because cerebral cortex has some degree of control over that skeletal muscle. But I can only do that until I piss off my brain, and then it won't let me do that anymore. So the higher brain centers can kind of override what the medulla oblongata and the pons are doing. And then the hypothalamus, which is sort of the emotional portion of the brain, also pain, those things can cause changes in breathing as well. So you can get so upset that you're, right? Or in pain, or in so much pain that you're doing that as well. So emotional centers or cognitive centers can kind of override what your pons and medulla oblongata are doing. But then, not surprisingly, your body is actually monitoring the chemical content of the blood to figure out when you need to breathe more or less. Now, we've talked about this before with monitoring circulation to figure out when you need to turn your heart rate up, right, or down, or when you need to do systemic vasoconstriction or vasodilation. So now what I'm talking about is that you are also going to be monitoring your chemical content of the blood, same sensory input, but this time I am going to use it to decide what's happening with the respiratory system. So not surprisingly, the things that you are going to be monitoring are going to be similar to things that you've looked at before. Okay, you are going to be monitoring the PCO2 of the blood, the pH of the blood, and the PO2 of the blood. So the PCO2 is interesting. So interestingly, with the respiratory system, more powerful than low oxygen is high carbon dioxide. In part because high carbon dioxide will, of course, change the pH, which has body-wide implications. So the most powerful respiratory stimulant when you are at rest is actually PCO2, not PO2. So let's look at kind of why that is. So the PCO2 of the blood. So let's talk about what's happening with the PCO2 of blood. Okay, so here's the deal with PCO2 of the blood. The carbon dioxide that is dissolved in the blood plasma. That stuff, if you carry a lot more than 7% of it, it will piss off your brain because here is what happens. Okay, so the bicarbonate ion cannot diffuse into the cerebrospinal fluid. Get out of there, you little bastards. So H plus can't diffuse into cerebrospinal fluid. Bicarbonate can't diffuse into cerebrospinal fluid, but carbon dioxide can and will. So as the carbon dioxide of your blood goes up, not the pH of your blood going down, but the carbon dioxide of your blood goes up, it will diffuse into cerebrospinal fluid. And as it diffuses into cerebrospinal fluid, then you will force this dadgum chemical reaction, CO2 plus H2O, carbonic anhydrase, carbonic acid dissociates. And this right here will piss off the chemoreceptors in your central nervous system, okay? And your brain does not play well with others. If you piss off your brain, it will shut down the whole damn thing. So basically, the PCO2 of the blood, if you increase the PCO2 of the blood, it will cause extra diffusion of carbon dioxide into CSF. CSF has terrible buffering systems, almost none really. And then the central chemoreceptors, because they're part of the central nervous system, will get activated in the medulla oblongata. They respond to low pH, and no matter what anything else is doing, that is going to make you breathe. It says there is too much carbon dioxide, you are pissing off your brain, breathe, I don't care what else you're doing. So low PCO2 in the blood is the most powerful respiratory stimulant. And then the pH of the blood. The pH of the blood is less powerful than this because, of course, the H plus itself doesn't diffuse into the brain. So blood pH really doesn't diffuse into the brain appreciably unless you've got some damage in your capillary endothelium. So blood H plus diffuses very poorly from the blood to the CSF and doesn't significantly affect the central chemoreceptors in the central nervous system. But what it will do is it will affect the peripheral chemoreceptors that you've already learned about. These peripheral chemoreceptors are in the aortic arch and the carotid sinus. This one's easier to piss off. So putting this back together with what we learned before, you remember that you were monitoring oxygen, carbon dioxide, and pH in your aortic arch and the carotid sinus. Last time we were using that sensory input to figure out whether to turn up or down the cardiovascular system pumping. Now we're going to use the same sensory input, only this time we are going to use it to figure out when to turn up or down ventilation. So the increase of H plus, if detected at the carotid sinus or the aortic arch, what those will do is that sensory information is going to be sent via a nerve that you don't really care about, the glossopharyngeal nerve. It's going to be sent via the glossopharyngeal nerve. It's going to be sent to the medulla oblongata and the pons, and then you are going to say, oh, low pH or increase of H plus in the blood at these locations sent, and it's going to say, oh, the pH of the blood is too low. What should I do? Should I breathe more or breathe less? Well, yeah, you should breathe more, right? Even if the low blood pH wasn't due to carbon dioxide, that's how the respiratory system is going to help with the pH of the blood. So basically, every low pH problem, I should say, with the respiratory system is dealt with by increasing ventilation, right? Even if it wasn't due to carbon dioxide. And then last but not least, last thing on this set of notes is, although it's very counterintuitive, the thing that is least able to change your ventilation rate out of carbon dioxide in the blood, pH in the blood, and NO2 in the blood, the thing of the three of those that is least robust at changing your ventilation rate is actually PO2. So the PO2 has very little impact on ventilation rate if detected in these same locations until the PO2 falls below about 60 millimeters of mercury in the aortic arch and the carotid sinus. When the PO2 falls here below about 60 millimeters of mercury, it will stimulate the peripheral chemoreceptors, these, and cause an increase in ventilation. But until it gets really, really low, it doesn't change ventilation rate. So when it gets really low, it will do it. So in order of importance to activate ventilation, this order: increase in PCO2, decrease in pH, decrease in PO2. Those are the three that will cause ventilation increases. Okay, I want to remind you again that this guy right here, this ARDS and how the coronavirus kills, this is going to be the basis of one of your bonus questions. I think it's really good to try to put together, and what I'm going to try to do is ask you to put together a little bit of what you've learned from the notes with what he's talking about. And of course, you'll get partial credit. I want you to try to get this information in. That'll be one of your two bonus questions that you have to choose from. You're only going to do one of the bonus questions. Okay, one more lecture and then we're done. Okay.